Hello class. So for this video I'm going to go over the labeling for the microscopes and cells. I know for one of the labs we got through both microscopes, so you might want to skip forward a little bit, but I'm going to do these first and then we'll get into the cells. I'm going to start with the microscope we worked with and then get back to the dissecting one. So up here you have the oculars, that's this box. Under the labeling or next to the labeling you probably want to put its function. So the oculars allow you to view your specimen, and it does a low amount of magnification, 10 times. This next thing is this wheel. See this rubber part on the outside of this metal? That wheel is the revolving nose piece, or just the nose piece. And it allows you to change your objective. This is an objective, so it's going to be this big box. Probably you want to put their colors and magnifications. But they're going to be the main lens that magnifies your specimen. And so red is scanning power, it is 4x. Yellow is low power, it's 10x. And blue is high power, it is 40x. When you find out total magnification, you multiply the eyepiece magnification, which is 10x, times the objective lens, which varies which one you're using. So again, for scanning it's 4, low is 10, and high is 40. So we'll multiply these together to get total magnification. At scanning power, the total mag is 40x. At low power, 10 times 10 is 100x. And at high power, 10 times 40 is 400x. There's also going to be another objective on our scopes that we don't use. It's either white or black. And it is the oil objective, and it requires microscope oil to use, or immersion oil, and we won't use it in this class. Anyway, so this little metal piece right here is the slide clamp, and it will hold your slide in place while you view it. This black platform right here is the stage, and the stage is where your specimen sits that you were looking at. You can see this little lens peeking through this slot. That is the condenser. But it also has the diaphragm. And remember, diaphragm has a weird spelling, diaphragm. Oh no. You might change the font size or something so that it all fits. Anyway, so that's your diaphragm and condenser. Stop it. And the diaphragm controls the amount of light that hits your specimen. The condenser focuses light on your specimen. This little box down here is the light source. It illuminates your specimen. And then there's this set of controls hanging off the stage. And these are the stage controls. And they allow you to move the stage around and therefore your slide around. So you can look at different parts of your slide, different parts of your specimen. This larger diameter wheel down here is the coarse focus, and the smaller diameter wheel is the fine focus. You're only going to use the coarse focus at 4x at scanning power. You'll use the fine power at 10 and 40x, the low and high power. But they both allow you to focus on your specimen. And then this, I believe, is the light switch. And it'll turn your light on and off. And anytime you stop using a scope, you want to turn that off. Alright. Now this term for the lab practical, it just means that if you get your specimen and focus at 4x, you can switch objectives and it'll be close. So you should only need to use the fine focus anyway when you're switching objectives, and that's what parfocal means. Alright, so this is a dissecting microscope. It allows you to look at 3D images, whereas the light microscope, it's mostly 2D. But this setup is pretty much the same. It's a little bit simpler. There's your oculars. They allow you to view your specimen. This is the stage and one of the light sources. So it will allow you to, it's where your specimen goes. This wheel is your focus, and there's only one focus for a dissecting microscope. And then this little switch changes your magnification. And it's usually between like 10 and 30 or 10 and 50. It kind of depends on the microscope. But that is it for the microscopes. Okay, so if you got through the microscopes, you want to start here. Again, we probably want to summarize their function as we label. So you can put it out to the side or maybe in the box. This is a plant cell. You can tell by sort of the geometric shape and the fact that it has multiple layers on the outside. And then this is an animal cell. Its shape is a little bit more round and free, and it only has one outer layer. So let's do the plant cell first. This whole big purple thing is the nucleus. The nucleus contains the DNA. This darker spot inside the nucleus is the nucleolus. So pay attention to spelling a little bit. So nucleolus pretty much means tiny nucleus, but it makes ribosomes. It makes the rRNA. This part of the blue stuff that looks very much like sheets, or sort of sections, that is the rough ER. It makes proteins. This blue stuff over here looks tubular, that is the smooth ER. It makes lipids. This large clear thing in the middle, that is a central vacuole, and it stores water. It helps the plant not burst when it's in hypotonic water. This thing with the green discs in them, they are chloroplasts, and they do photosynthesis. So with sunlight and water, they can make sugars. They can make carbohydrates. This darker tan layer goes all the way around, except they cut out a piece here. That is the cell wall, and it is for protection, and it creates the cell shape. This white or clearish part lines the inside of it, and that is the cell membrane or plasma membrane. It controls what goes in and out of the cell. This little orange thing is the mitochondria, and it is the powerhouse of the cell. It makes ATP. These little purple, well, they're sort of pink. The pink spheres, They are called vesicles. And they are little packets of membrane for transport. So they can transport things anywhere in the cell. And then these pink sheets, that is the Golgi apparatus, and it's in charge of transportation, so it does receiving and shipping and packaging, but also in the Golgi you can modify stuff. That was the rough VR, and then this is pointing at these little dots. Those are free ribosomes, and they also make protein. So that's the plant cell. Let's do the animal cell real quick. A lot of it will be the same. The animal cell does not have a cell wall. The animal cell does not have a central vacuole, but they can have normal ones. And the animal cell does not have chloroplasts. One thing the animal cell has are these centrioles, right here, centrioles. That's one thing the animal cell has that most plant cells don't. And the centrioles move the cytoskeleton. They control the cytoskeleton in the cell. Speaking of cytoskeleton, these purple tubes, and you can barely see these orange ones, those are all components of the cytoskeleton. They determine cell shape and allow cell movement. This purple thing is probably a lysosome. To lyse means to break. These lysosomes are much like a stomach. They're very acidic, and they have hydrolytic enzymes, and they can digest things. And these things can be bad things like bacteria, or they can digest stuff for recycling purposes. This clearish white outer layer is the cell membrane, the plasma membrane. It controls what goes in and out of the cell. This blue tubular stuff, again, is the smooth endoplasmic reticulum, and it makes lipids. This whole purple thing is the nucleus. It holds the DNA. This dark spot within the nucleus, that's the nucleolus. It makes ribosomes. Alright, so I think this one, even though it's not exact, are pointing at these orange dots. And those are the ribosomes, and they make proteins. I think this one is pointing at these blue sheets with the ribosomes on it, which is the rough endoplasmic reticulum. And it also makes proteins. This pink stuff is the Golgi apparatus. Again, it does shipping, receiving, packaging, but it also modifies stuff. And this pink sphere is a vesicle, and that is for transport within the cell and maybe to the cell surface, so out of the cell. This orange thing is a mitochondria, and they make ATP. So that's everything you needed to label. Again, you need to know the functions of these as well as being able to identify them for the exam.